This is episode 281 of the Stem Cell Podcast, Fertility Treatments, with doctors Dina Radenkovic and Bruna Paulsen. Hey, everybody. This is Dr. Dalen James and Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the podcast, rate us and leave a review. We're always looking for feedback on how we can improve things and for suggestions on guests. Today, we have doctors Dina Radenkovic and Bruna Paulsen, who are both from Gameto. They're on the podcast to talk about their work on reproductive medicine. Really exciting stuff there. Can't wait for that chat. We've also got our usual roundup on recent highlights in stem cell news. That's coming right up.
But first, interested in exploring the applications of organoid cultures? Get a free wall chart showing how organoids are used as model systems to study infectious disease, cancer, congenital disorders, and tissue regeneration. To learn more, visit stemcell.com/organoids-modeling-wall chart. We're going to start things off with a paper in Nature, came out at the end of October. This is coming from Cameron McAlpine at Mount Sinai in your neck of the woods up there at the Cardiovascular Research Institute. First off, for here is Pacific Hyun. This is a unique study looking at heart attacks, myocardial infarction, and I guess a unique aspect of how you might be able to improve or mitigate some of the damage that's done after a heart attack, just because of sleep, right? Just using sleep as a way to limit cardiac inflammation and damage. So that's the title of the paper. Myocardial infarction augments sleep to limit cardiac inflammation and damage. It's almost like a self-protective mechanism that's built into the body after such a dramatic and severe event, such as myocardial infarction. And, everybody knows about the importance of sleep. It's important for cardiovascular health in general. You got to get your sleep, right? I wish I got more sleep. But the circuits that are actually connecting cardiovascular pathology, such as in the context of myocardial infarction and sleep, are incompletely understood. And it's not super clear whether cardiac injury can influence sleep or vice versa, and whether sleep-mediated neural outputs can actually contribute to healing of the heart after something like a myocardial infarction, and also the role that inflammation has here. So this is a really cool study that was building on a mouse model, a myocardial infarction mouse model, which is pretty common in the field. I mean, I've used it in the past a while back, and they showed that monocytes, this subset of immune cells, are actively recruited to the brain after after myocardial infarction, to actually improve sleep, which in turn suppresses the sympathetic outflow to the heart. That limits inflammation and actually ultimately promotes healing of the heart. Now, we're not talking about regeneration because, as we know, the human heart doesn't regenerate itself, but we're talking more about limiting inflammation because there's that big problem of the fibrotic scar and all the downstream effects after a myocardial infarction, right? After having a heart attack, your cardiac function is impaired for oftentimes the rest of your life. And so ways to mitigate that damage that's done right after the MI is useful. I mean, this is a really powerful mechanistic study in that way. So after the MI, the microglia actually rapidly recruits circulating monocytes to the brain's thalamic lateral posterior nucleus, or LPN, via the choroid plexus, where they're apparently reprogrammed to generate tumor necrosis factor, or TNF. Okay, we've talked about TNF in the past, and in this thalamic LPN, these monocytes, the TNF from the monocytes actually engages another signaling pathway that ultimately activates the glutamatergic neurons to increase slow wave sleep pressure and abundance. So if you disrupt sleep after MI, it'll actually worsen cardiac function. Pretty sort of straightforward in that way, I guess, right? And that's going to decrease heart rate variability, cause spontaneous ventricular tachycardia, A lot of downstream impacts here. And after MI, disrupting or curtailing sleep by manipulating that glutamatergic TNF signaling can actually increase cardiac sympathetic input, which signals through, as you might expect, beta-adrenergic signaling, and this is through macrophages. So this is a complex series of interactions here that's happening between the heart and the immune system, and that everything I just said was in a mouse model, which you would say, okay, fine, great, show that in a mouse model, but of course you have to demonstrate in some capacity that this exists in a human context, and you can't do those ultra mechanistic studies, of course, but you can look at data sets of people who have had myocardial infarction. So that's exactly what they did. So they showed that poor sleep in the weeks following an acute coronary syndrome myocardial infarction actually increases the susceptibility to secondary cardiac events and reduces the heart's functional recovery and it has an increase in inflammation as well. And so in parallel, insufficient sleep in humans reprograms this beta-adrenergic receptor signaling, expressing monocytes towards a chemotactic phenotype, enhancing their migratory capacity. And ultimately, all this data, this mouse data plus this human data, is showing that there is a cardiogenic regulation of sleep after heart injury, which can restrict the sympathetic input of the heart, ultimately limiting inflammation and damage. So if you have a heart attack, you got to get your sleep. But I think it's not just in the context of heart attacks. You have to just get your sleep in general. We know there's so much we actually don't know about how sleep impacts the body. And we know it has this regenerative capacity, but the mechanisms of that are not well understood. And so, at least in this very specific context of myocardial infarction, showing that you got to get your sleep and it's ultimately going to help your heart feel maybe a little bit better.
Yeah, this is such a neat study, and as you just were alluding to, there's this idea of sleep. It's not just about like a regenerative potential of a therapy, but like creating a non-inflammatory. It's like taking away the insult can be as uh, you know uh, reparative as a regenerative impetus. So conceptually, a real interesting thing there. The study in total, as you said, super complex in terms of mechanism, how you link one thing to the next to converge on sleep, which is like a real simple idea, right? It's like, oh yeah, of course, you just had a heart attack, take a nap. But what's so kind of it totally makes sense, but at the same time, I'm so confused and perplexed because I'm thinking, one, is this generalizable, right? Is this something that is just about injury of any tissue? Because if it is specific to the heart, then that's a bit of a twist, right? Because like I guess MI is relatively survivable in the wild if evolution, maybe it's some, heart injury in lower organisms that's conserved up to humans. But I don't know a lot of people in the caveman days that were having an MI and then just going about their life. But maybe they were, right? I guess that's one implication here that MI is not irreconcilable with survival in a natural context. At least evolution didn't say so. But a little bit, I want to take it up with Mother Nature here and say if you went Rambo on this mechanism here just to get people to take a nap after MI, like, couldn't you have slipped a little something in there in terms of a regenerative progenitor or something that could have maybe bypassed the scar or mitigated that? You did all this work to get people into bed. What about, an adult stem cell, Mama Nature?
Well, first of all, I think back in the Stone Age, I think folks had other problems to worry about except for other than myocardial infarction, like getting eaten by cheetahs or something. Hey, it's an interesting reflection on the evolution of a mechanism like this. As we've talked about ad nauseum here on the show, other animals, lower vertebrates like zebrafish, axolotl, have innate regenerative capacities in their hearts, so maybe they don't have to utilize sleep in this context. Those hearts can just fix themselves after something like myocardial infarction. But yeah, I think you're alluding to something here that this is a really really intricate mechanism that they're trying to flesh out. And I like you would be really interested in seeing how widely applicable something like this is. I mean, sleep is good in general. Just sleep is good, right?
Yeah, I could use some sleep, partner. It's been some time since I had a perfect night's sleep, and now you know how that is with a young child, another on the way. Bumming. Anyway, moving on to a bit of a segue here. You know, one of the ways we talked about on the show that we've tried, not we, but science has tried to address MI is using CAR T, right? CAR T for fibrosis. That's a little bit, I don't want to say far fetched, but a little bit, on the horizon in terms of whether it can be practical. But CAR T, amongst other things, has revolutionized the treatment of cancer, right? Um, Truly revolutionized. A new day for a lot of treatments that weren't responsive. Um, and one of the challenges there is that it's still really expensive. So not really widely applied because it's really labor intensive. You got to go autologous. Of course, we've all been going after this holy grail of iPS cell, hematopoietic stem cell, which would offer a solution, but also just generating lymphoid cells from iPS cells, which has been done. Um, but generally, the iPS-derived T cells have this immature, kind of an innate like T cell, which is not as functional as the mature peripheral blood-derived T cells that you use for the autologous CAR T therapy, right? So we're still stuck with the autologous, still revolutionary, but not exactly cheap, right? And one of the ways that George Daly, who God love him, he's been hammering away at the hematopoietic stem cell, lymphoid, everything hematopoietic from pluripotent stem cells from day zero. Still is, and this is a illustration here, this cell stem cell paper of the incremental progress on a grand scale. And it's building on a previous study where he showed that if you repress this EZH1 um, a gene, you can facilitate production of lymphoid progenitors. So EZH1 is this epigenetic modifier. They also uh, developed this stroma-free protocol. So stroma has also been an issue always for like clinical practicality. So they've really refined incrementally this many studies that I'm not even going to mention here, but using uh, that stroma-free protocol, they show that if you knock down EZH1, you could get mature T cells, right, from iPS cells. They call these the easy T cells. And these look a lot like the T cells that are in the peripheral blood, and they show this robust anti-tumor activity when you add the CAR, in this case, anti-CD19 CAR, which is widely used for B cell based cancers. But the problem there is that you have to use this shRNA-based knockdown of EZH1 to get these cells, which is not really practical in the clinical paradigm as it stands, given the current approaches to knockdown. And the small molecules that you could use to knock down EZH1, they all also knock down, block EZH2. So both those coupled has deleterious consequences for the cells. So here, they built on those two studies, screening again in the stroma-free protocol, looking for small molecules here from a library of 150 of these epigenetic modifiers. Small molecules repress epigenetic related factors, and they found this repressor of G9A/GLP, which is this H3K9 directed histone methyltransferase. They show that if you inhibit this, you can skew fates toward lymphoid lineage, and in the context of zebrafish, which I think was just an opportunity to get Lenz on the paper, he's second to last here, so him and George could have fun together doing experiments again. So they showed in the context of zebrafish embryogenesis that it's skewed to lymphoid. uh, hematopoiesis, so in vivo. So that was important, though. I joke, but it was important to show that this is not something that's like unique or hack. It's conserved all the way down to the zebrafish, and then, of course, they show that if they use this inhibitor of G9-AGLP, they can get iPS-derived T cells that, again, look like these mature autologous peripheral blood-derived T cells used for CAR T therapy. And when they load them with the chimeric antigen receptors, they show an enhanced effector and function, and this anti-tumor activity in this xenograft model. So, as I said, it's incremental, but like on a grand scale, this is a major problem and has really tremendous potential to address a wide swath of diseases. Here we're talking about cancer in terms of just increasing the scope of treatment, increasing access. But as I started by saying that, the idea of using CAR T for fibrosis, for solid tumors, not just hematologic, for autoimmune disease. So this we're talking about like a tool that could be revolutionary across many disease sectors. So this was a major step forward in getting iPS cells to generate lymphoid cells that look like these robust peripheral blood derived T cells used for CAR T in the present day. And we're talking like unlimited amounts. So pretty big deal.
Yeah, good stuff in the Daily Lab. Been working on this for a long time now, them and many others in this field. I think, the economics of the iPSC derived CAR T is still something that needs to be figured out. Initially there was the autologous approach, but, generating iPSCs from someone's own skin and blood and then using those ultimately for therapy, it's perhaps somewhat cost prohibitive. So maybe more of like an off the shelf approach is the way to go. But regardless of the economics, I think this is a really important study that pushes forward the iPSC derived T cell field. I think it's one of a few papers that we've been chatting about here on the show recently when we're in the realm of using pluripotent stem cells to derive blood lineages. There's still that holy grail that we've alluded to a lot of getting bona fide hematopoietic stem cells. I think the Elefante group had that Nature Biotech paper a little while ago, and we talked about that. And this is, again, in that same realm of how do you make these blood cells derived from iPSCs truly bona fide, mature, and ultimately doing what they're supposed to be doing. And I think we're getting close. And this is just another example of that.
Yeah, and I think, to his credit, of course, Daly and his group and many who've come out of that lab, of course, no one has ever given up. I've given up, I'll be honest, but not everybody has given up on hematopoietic stem cell here. But I think what we've learned as a community is that you can get past the stem cell. I mean, I guess that's the real challenge, is being able to create the appropriate niche to cradle that stem cell through that transitional phase, and isolate it from the pro differentiation signals so that you can actually get it to stick, pull it out of there. And yeah, the Elefante group is a great example of how, like, we may have gotten long-term engraftable, although serially transplantable, let's see, but long-term engraftable cells. But like, can we get the cell? Can we look at it and expand it in a self-renewing state and then transfer it? Like, no, I don't think anyone's got there. I think being able to truly isolate and expand the cell is a challenge that may never be met, but I'll never say never there. I think it will. But in this case, I think it shows the utility of just going to a different endpoint, being able to get these cells to flux through this, stem progenitor state toward lymphoid, but getting them, unlike, and it's a challenge facing, you've described it many times, maturation, right? To getting a correlate to the adult, the tool that's going to be useful either therapeutically or, in this case, like as a therapeutic in itself, like for targeting. And that's why I really don't think you can overstate the advance here. I think that we have now a ready for prime time tool that is off the shelf capable in the context of these hypoimmune ES cell lines, that is, has potential to address so much unmet medical need that I'm really curious to see where you go from here in terms of trials. Like I want to know what more you got to do to get these things into trial. So we'll be keeping a close eye on this for sure.
Yeah, that's for sure. And it's not just the Daily Group. Like I said, many other folks in this field are working on this, and so really just a matter of time. Moving on to another cell stem cell paper. This is coming from my former lab at Stanford. None other than Joe Wu, who we had on the show a while back. Their lab has worked on this idea of doxorubicin induced cardiotoxicity, which is actually kind of what my lab works on too, as an acolyte of Joe Wu, I suppose. The general concept is that cancer drugs can do damage to the heart. This is a huge problem, and there's some statistics here that are showing that there's going to be more than 26 million cancer survivors in the US by 2040. And for a number of these survivors, unfortunately, they're going to be impacted by this off-target effect of their therapy, which is, damage that's done to their heart, whether directly or indirectly. And this is, like I said, the Wu Lab has been exploring this for more than 10 years now. I've actually published a couple of papers in this area as well when I was in Joe's lab. And this is in the same realm. The title here is CRISPR A and CRISPR I screens and human iPSC cardiomyocytes are identifying a glycolytic activation as a druggable target for doxorubicin-induced cardiotoxicity. About 10 years ago, I think the initial paper on using iPS cardiomyocytes for examining drug cardiotoxicity was published in Joe's lab by Paul Burridge, who we've had here on the show and is a mentor of mine. And this is one of the follow-ups that's been coming out of the Joe Wu lab in that area, again focusing on doxorubicin-induced cardiotoxicity, but using this new CRISPR-based tool of CRISPR-I, CRISPR inhibition, CRISPR activation screens to look at some new targets potentially for alleviating doxorubicin-induced cardiotoxicity. So doxorubicin is this, hallmark cancer drug that's been around forever. But forever, it's been known for its ability to cause life-threatening cardiotoxic side effects, heart failure, arrhythmias, cardiomyocyte death. So it's called the Red Devil for a reason. And here they're presenting an integrated drug discovery pipeline combining iPSCs, iPSC cardiomyocytes, CRISPR-I, CRISPR-A, bidirectional pooled screens, real Joe Wu Lab approach. They go really big on the screening here. The small molecule screen to identify some therapeutic targets that can actually mitigate doxorubicin-induced cardiotoxicity without compromising its on-target effects. That's really important. So for whatever cancer drug you're designing, you want it to be effective on target. You want it to eliminate the cancer, of course, breast cancer, whatever, but you want to reduce its off-target impacts. So the screens here, they revealed several previously unreported candidate genes contributing to doxorubicin-induced cardiotoxicity, including something they identified here, CA12 or carbonic anhydrase 12. They inhibited the CA12 and found that, surprise, surprise, it protected iPS cardiomyocytes against doxorubicin cardiotoxicity, improved their cell survival, sarcomeric integrity, contractile function, calcium imaging, all these properties that are critical for keeping cardiomyocytes happy. They had another compound called this indisulam, a CA12 antagonist, which was effectively attenuating or alleviating doxorubicin-induced cardiotoxicity in iPS cardiomyocytes. I think this is not a brand new drug. It's a repurposed FDA-approved drug previously, which makes its new indication perhaps a little bit easier to attain, right? It's already gone through the rigors of FDA approval once, so you just got to repurpose it to a new indication. So this drug was able to attenuate doxorubicin-induced cardiotoxicity in a bunch of different model systems that they have in the Wu lab, including engineered heart tissue. Of course, they had animal models as well, showed that the indisulam could do its thing there. And then mechanistically, they dove into it, figuring out that doxorubicin-induced CA12 actually potentiated a glycolytic activation in cardiomyocytes, and that's how it contributed to DIC, doxorubicin-induced cardiotoxicity, interfering with metabolism and various other cell functions. So this is a really great nuts to bolts Joe Lab kind of style paper. You got the screening, you got the iPS cardiomyocytes, you got the repurposing of an existing FDA approved compound, and ultimately perhaps another target, another target that they found here in this indicilam that could alleviate doxorubicin cardiotoxicity. I know Paul Burge is working on similar approaches as well. They're just building towards this roadmap for future drug discovery efforts, potentially leading to more targeted therapies. That's the goal here. Targeted therapies in cancer, but targeted therapies that have minimal off-target cardiotoxicities. So shout out again to Joe and his team. First author here is somebody who I've actually worked with in the past, and Shun Liu. Shout out to the whole team, and yet another great study in this area of cardio oncology.
Yeah, you said it. Woo Lab doing Woo Lab things. And another candidate. Yeah, I'm looking at my AI here. Tells me, I think there's some irony here that Indicilam, it's in phase 2 trial now in the US and Europe. Good toxicity profile. The clinical response is modest, but toxicity profile, that's solid. But the irony is that it's this anti-cancer drug for melanoma and bloodborne cancers, including leukemia. It's cell cycle inhibitor. So I guess that's the treatment. We got to give you one anti-cancer drug for your cancer, and then we got to give you another anti-cancer drug to protect your heart from the other anti-cancer drug. But like as you said, a favorable profile from the FDA speaks to a short runway to seeing how this works in actual human trials. Again, another one of these off target, or at least off initial target applications of pluripotent stem cells, in this case, tox modeling that looks to bear real fruit in terms of like moving the clinical needle.
Yeah, it's a low-hanging fruit, like what you're alluding to here. If something's already been approved or is in late phase trials already, it's easier just to kind of shift it around and, like you're saying, a shorter runway for approval for this other indication. I think this is one of the, um, I think, big victories in iPS cardiomyocytes and iPSCs in general, and identifying novel compounds, therapeutic compounds that can be repurposed into clinical trials. Um, the cell therapy side of it, of course, we've talked about a lot here on the show, and that's happening. But I think this is a victory that's already established. There are compounds that are being actively used, and I can think of some other compounds in the realm of heart failure. Mavacamten is one, for example, has been FDA approved and is being used in hospitals across the country. And some of those initial screens to identify that mavacamten actually can improve cardiac function, those screens were done in iPS cardiomyocytes. So those victories have already been established, at least for the in vitro side of the field. So yeah, really great stuff from the Woo Lab.
Yes, the clinical impact. That's what we talk about a lot on this show, and we love it, the way it's moving. But at heart, I say it all the time. I'm a developmental biologist, and part of the wonder for me, I think I speak for a lot of scientists who use stem cells as a tool, is that you're finally able to to see something you could only ever imagine, right? This idea of the complexity of differentiation, cells in space, but also in time, in this constantly evolving, molecular spatiotemporal landscape with multiple axes. It's really miraculous every single time. And that's part of what drew me to the work. And I think a lot of developmental biologists have been fascinated by the interplay of not just the cells in space, but in time, and the link of expansion of a cellular cohort, cell division, and the rate of cell division, and differentiation, right? Because especially in lower organisms, all these differentiation events happen at these very prototypic time points. And that is held up, right? The Carnegie stages is pretty reliable. At the single cell level, of course, there's some variability, but things happen in a certain order and a certain time. But the question I think that remains is that while cell differentiation um, can result in different cell cycle machinery that dictates a different rate of cell division. We've all seen it. You differentiate to a certain lineage, it pops. A certain one is slow, right? I mean, you could tell me cardiomyocytes, they're not at the mature state. They're not really moving in terms of the cell division. But the reciprocal question remains, I think, a little bit murky, right? Whether cell division or how tightly cell division is coupled to cellular differentiation, right? There's a lot of examples where differentiation events are dependent on like cell division. For example, there's binding of a transcription factor that only happens during S phase, right? So that's like a cell division dependent event, or you'll get accumulation of transcription factors because cell cycle will increase in time. So it allows for these transcription factors to accumulate. This is what happens in myeloid cells with transcription factor P1. Also getting like a critical nucleocytoplasmic ratio, which only happens after a certain amount of divisions, or asymmetric partitioning of cellular components to dictate like asymmetric fate dictation, right? Like all these concepts. So it all seems there, right? But the converse is also true. You can take embryos across many species and do cell cycle arrest, and you'll still get morphogenesis. You'll still get, even the cells will stop dividing, but they'll still move around and form structures, and they'll still differentiate even on a cell intrinsic level, take on new fates, right? So it's known that timing is important, but it's also known that the differentiation morphogenesis can happen kind of outside of timing. But the degree to which that happens across the many lineages and cell types within an embryo is really unknown. It's just, both things are true. But for certain cell types and certain niches, certain tissues, we don't know on a more granular level. And that's what seeded this question and this effort from Alon Klein, who's at Harvard Med School, using zebrafish. They use this kind of extra uh, cell cycle inhibition, right? They were able to freeze, like with DNA replication inhibitors, hydroxyurea, some other things, cocktail. They were able to show that cell division is dispensable for differentiation um, across all embryonic tissues, starting from early gastrulation to the end of segmentation in zebrafish. So it was murky, right? Both things are true, but here it's just, yes, it's a general principle that the dependence on cell division is not really there. There is a relationship, of course, that there is a slowing of differentiation in some cell types. Also, globally across all cells, there's this stress response, kind of like a DNA damage response, which you can imagine a cell is kind of confused, and maybe the pile up of certain, I don't know, factors in the cell that get evenly distributed division, who knows? But you get this stress response that's a global. But the degree to which it affects morphogenesis and differentiation varies across tissues, but mostly it doesn't. And what it shows, I mean, fundamentally, using this and the technique, of course, we talked last episode about this zebrafish hub or zebra hub, whatever it's called, uh, which was just looking in normal development at single cell, single embryo seq. Here they use a similar approach, except in the context of these embryo-wide perturbations. So this is kind of like an atlas of this phenomena that they're describing. And while the differentiation, as I said, is moderately affected. It shows that it's not dependent on cell cycle progression, but like properties like the total numbers of cells are tuned by its different division rates. So a really great, I think, fundamental basic story that has implications for our own work, because we talk so much about developmental timing and maturation, and I think this further reinforces this idea that you can get kind of outside of the timing of differentiation. I don't want to say you can bypass it altogether, but you can hack it, so to speak, and get to the endpoints you want, maybe by indirect routes. So a basic conceptual advance here that I think has a lot of application to our own work in stem cell differentiation.
I mean, that's pretty much exactly what we're doing whenever we're doing an in vitro diff. An in vitro diff is a hack, right? We are hacking embryonic development and trying to recapitulate it in a way that suits our needs to make whatever rudimentary cell type you want in vitro. It's not perfect, certainly, but I think this concept of using the embryo and the developmental processes in the embryo to fine tune differentiation ultimately is, I think, something that a lot of people are working on. This is also just another reflection of the power of the zebrafish as a model system. I mean, this is a study that's uniquely enabled by the fish, by the zebrafish, because of its great characteristics for genetic manipulation, optical imaging, and so forth. And I think this coupled with things like Zebra Hub, Zebrafish Hub, whatever you want to call it, will help us understand zebrafish biology to an exquisite level, to a level that hasn't been done before. So I'm excited for what this means. The evolutionary part of it is something that I'm also really interested in. Is for some of these mechanisms, how well conserved are they across different species? Because as we know, zebrafish has a very unique capacity for, say, regeneration, for example, but its developmental timeline is also unique in that way too, right?
Yeah, and as you're saying it, I realize this is for once. We talk about zebrafish, the power of zebrafish, and here it's not what we usually talk about, which is what an amazing model it is for imaging and live cell imaging and tracking, lineage tracing, etc. Like, this isn't a paper. You're not going here for the images, for the micrographs. You're going here for the atlas, and I'm glad that you mentioned that, because this, with the zebrafish hub, I think is elevating this concept of like a whole embryo or whole organismal single cell seq. And I think it's a little bit beyond what we're capable of doing in mammals, but it's something that is going to, I think, enable a lot of insight into these more fundamental processes. And yes, as you said, very incisively, I might say, Arun, is that the question of conservation is key. And I think that maybe on a smaller scale, not on a whole embryo or whole organismal atlas level, but on maybe like a tissue level, we'll be able to ask these questions, looking at mouse, of course, and but also even human fetal tissue and stem cells. So you think we're at the end of the days of the single cell, but it's really just the beginning here. Now we're just like ramping up the deployment to more and more sophisticated applications. And I mean, get yourself a bioinformatician or get familiar with Seurat if you have not already, because you're going to need it, my friend. Moving on to the interview, we talking to a couple of scientists who have a lot to say about the basics of development. They're going down to like the single zygote is their focal point. Before we get there, though, I have a quick message from STEMCELL Technologies. Are you looking for a robust and versatile system for your female reproductive biology research? With stem cell technologies, Gynecult fallopian tube organoid medium, you can generate highly representative and functional fallopian tube organoids in just 10 days. From fertility studies to ovarian cancer research, Gynecult gives you full control over hormone levels and delivers consistent results to power your breakthrough discoveries. To learn more, visit stemcell.com/gynecult. That's GYNECULT -FTOM. All right, everybody. on today's episode, we have what I would call a very special duo here, representing the point of the spear, you could say, of clinical translation of stem cell science, representing Gameto. We have co-founder and CEO, Dr. Dina Radenkovic, also VP of Manufacturing and Therapeutic Development, Dr. Bruna Paulsen. So Dina is a medical doctor and a healthcare entrepreneur. Prior to her role as co-founder and CEO of Gameto, she was a partner at Salt Biofund and had research posts at King's College London and the Buck Institute for Research on Aging. She qualified with a dual degree in medicine and physiology from UCL Medical School, awarded the best overall performance in physiology BSc. She did a residency at St. Thomas Hospital in London. Bruna did her postdoctoral research at Paola Arlada's lab in Harvard, a friend of the show, where she used human brain organoids to understand the molecular and cellular basis of autism spectrum disorder. Really cool work. I remember, I think we covered that in the show. Her interest in human pluripotent stem cells started when she was an undergraduate student at the Federal Universidad. I didn't need to do that, but I'd throw it in there. Of Rio de Janeiro, where she pioneered the work on reprogramming iPSCs as a strategy to model schizophrenia using patient-derived cells. Doctors, welcome to the show. Thank you so much for joining us.
Thank you so much. It's a pleasure.
Thanks for having us.
Yeah, welcome. And like Dalen said, this is a really unique episode. I think most of our guests have been basic academic stem cell biologists, many of whom actually work with induced pluripotent stem cells, iPSCs. But as we'll discuss in a little bit, this is a really exciting time for the iPS field because of the translational and the clinical potential associated with these cells, right? I mean, there's a ton of, not a ton, but there's a number of early phase iPS clinical trials underway, for example, with really sound science as the foundation. That's really important. Using these cells for different applications like treating neurodegenerative diseases, restoring cardiac function after myocardial infarction, and so on. Now, Gameto is uniquely positioned in that broader scope of companies because you're a biotech startup also utilizing iPS technology, but in the context of reproductive medicine. So to start things off, Dina, as the co-founder and CEO of Gameto, why don't you actually start off by giving us your kind of broad overview, or in startup speak, your elevator pitch of what Gameto is all about?
Thank you so much, and thanks for having us again today. We are big fans of the podcast, and when we initially searched, Bruna was like, "Well, it's mostly academic groups," and I was like, "Well, Bruna, we should reach out because now, if Nobel prizes are given in startups and to companies, like there's great science happening in companies, not just in academia. So maybe there's something interesting to share." So we really appreciate the invite. Gameto is, as you mentioned, one of the very few companies that uses the advances of cell engineering, but in women's health, with the lead asset focused on reproductive medicine, in vitro fertilization in particular. Women's health has historically been a neglected, underfunded area from all sorts of reasons. And we really want to apply this cutting edge science to this underserved field where we have so many common conditions that don't even have first line treatment. And pretty much everything is treated with hormones in one way or the other. So Gameto essentially leverages the cell engineering platform we've developed with the sponsor research agreement with Joe Church's lab at the Harvard Medical School and the Wyss Institute, where we convert iPS into different cells of the female reproductive system. We build these ovaries or synthetic ovaries in a dish, and then we use it to derive programs. Our lead asset is essentially an iPS-derived engineered ovarian support cell line that we use as an effective cell therapy in a dish. So we use it to mature eggs outside of the body. Currently, in vitro fertilization and egg freezing process, the way it works, and this process hasn't changed since the inception of IVF 46 years ago, a woman undergoes about 10 to 14 days of hormonal injections. These injections act on women's own support cells in the ovary. So we're giving systemic medication in order to kind of act on one cell type in the ovary to try and mature the eggs. And then we take these eggs out, and then the eggs can be frozen or fertilized. What we say is, why treat the woman for so long and cause all of the systemic side effects when you can treat her eggs? So we are essentially in a Fertilo cycle, which is the name of our lead program. A woman would undergo short and abbreviated stimulation of about 2 to 3 days with one injection per day. And then the eggs would be taken out, and then they would be kept in the co-culture, to which Fertilo is added, and we can essentially mimic a young follicle in a dish. So the final step of maturation happens outside of the body. That concept is called in vitro maturation. And then we shorten something that's 2 weeks long, incredibly difficult on a woman's body, both physically, mentally, financially as well, right? Like there are loads of hormone medications that need to be purchased, and into something that is hopefully a more accessible and a safer solution as we aspire it to be.
Thank you for that synthesis. I mean, that I think touches on many facets. One, first of all, as you led, the underserved and big unmet need of women's health is really near and dear to my heart and my research focus. And I love that this is something that is meant to reach it's very far reaching, right? This is meant to meet a need that is pervasive. And as Arun mentioned, there's a lot of these trials in clinical trials of cell-based research that are being conducted ongoing right now. It's very exciting. But the narrative there is, I think, a little bit more, I don't want to call it cut and dried, but it's more familiar, right? In terms of like you have a cell replacement therapy for a degenerative disease or injury, whereas this most advanced indication, as you described, is a co-culture system, right, to restore that native physiological microenvironment for oocyte maturation. So, Bruna, I'll put this to you. Like, in terms of like the apparatus that you need to put together to push this through, how does this differ, like specifically regulatory wise, from those other more familiar clinical trials? I mean, we talked to Blue Rock, and we've described a lot of these studies in early days. And this is a bit different, I would guess. Does like the lack of cell engraftment make for easier threshold vis-a-vis like safety risk? And in terms of like benefit, what are the targets? Like with degenerative disease, it's like you restore function to X degree to the normal or to the whatever apex or standard. Here, what do you have to measure in terms of demonstrating a benefit?
Yeah, so in terms of how, let me start with the first part of your question, like how this differ from the other clinical trials that are ongoing. We are still based on pluripotent stem cells, as most of them. We are internally taking this to the highest quality. So the way we are developing Fertilo is using the same quality that most companies are using to develop their cell therapies. Even if they are putting in the brain and the body of the patient, we are keeping our in vitro, but we are still developing with the same quality. With that said, Fertilo, in terms of differences, we are not treating a patient, we are treating one single cell. Our treatment is also way more abbreviated, and the cells of Fertilo will never go closer to that patient. So indeed, we are a lot more the risk than most therapies that are going to be forever, perhaps in the brain of that patient. But as I mentioned, we are still trying to keep to the same grade, same degree of quality and consistency of how we are making this product. And with regards to your second question, the way we are measuring efficiency of Fertilo is by looking at first mature eggs, and then euploid blastocysts, which is something that we have demonstrated in our study that Fertilo leads to higher rates of euploid blastocysts, and ultimately pregnancies and live births. So in comparison with other IVM methods that are currently in the clinics.
If I was to add here, I mean, I think there's an interesting analogy of degeneration and restoration of function. Personally, I see that this area of cell engineering is the most exciting area of biomedical research currently happening, because and it comes maybe perhaps to some of my research in the past, which was focused on kind of diseases of aging. But you could argue that the ovary is an organ that loses function much earlier than the rest of the woman's life. If you think about it, and if you look at the kidney, for example, like you have the glomerula as the unit of the function, right? And if you live long enough, you're going to start losing that glomeruli. And I remember if you look at clinically a patient, EGFR, right, the measure, you would look at the age, and if they're below 60, even if they're healthy, they would have a lower EGFR, right? So there's a loss of function of the organ, but it's synchronized with like the loss of function of the other organs in the aging process. With the ovary, you're talking about the fact that it's not synchronized with the aging of the rest of the human body. So actually, women experience a decline in ovarian function, whether you're going to call it aging or not, but it's a definite decline in organ function already in the mid-30s when they experience decline in egg quality and egg quantity. And then later on, so, women spend maybe about 2/3 of their life with increased life expectancy in an area of subfertility and infertility. And then we have the total decline of follicles, and then we experience menopause. And then again, with the increased life expectancy, we spend about half of our life in the stage of poor health with menopause. So actually, I would argue that, we're not that dissimilar. We're restoring ovarian function with cells. However, we're doing it we're attempting to do it, to be more exact, in a more, in an easier way, right? Instead of kind of like, if it's missing, replace, and you put it inside, we were saying, well, actually, there's still so many limits of cell therapies, with issues of immunogenicity and others. So given that this is already an in vitro use, why can't we have an in vitro application? And given that, we are really like the loners here in the world, like an IPS company in women's health, in IVF, right? Like, as you mentioned, we're very much kind of the pioneers and the first movers. We've said, why don't we focus on the first use of this technology to be in vitro? Because it's an easier scientifically. There are so many barriers of cell therapy, and Bruna can talk about, for example, the barrier of manufacturing at scale. We're not treating billions of cells, we're treating like 10 eggs. So the scale of the doses, the manufacturing, the cost, the issue of immune rejection, all of these things are really removed when you're treating 10 cumulocyte oocyte complexes. So it is like a first application, but really the broader mission of our company is that we use cell engineering to restore ovarian function and remove the phenotypes of a decline of ovarian function, which are infertility and menopause.
It's a great argument. I like your approach. You got me convinced. But
Thank you.
let's dive a little bit deeper into the weeds of the science. Like we started doing this a little bit already. We're of course a show for stem cell biologists by stem cell biologists, and as avid listeners of our show know, we're different from a lot of like more general science podcasts because we like to go like what we're already doing here. We're going deep into the technical papers and into the science. And Bruna, you said it yourself. You're a listener of the show. Thank you for listening. And you've done your postdoc with former podcast guest Paula Arlada, published a big time Nature paper on cortical organoids while you were there in the lab. So we're going to put you on the spot. We're going to ask you to take us through the details of some of the foundational papers actually driving the Gameto pipeline. So let's start with that, basic science eLife paper that you alluded to, Dina, from the Church Lab, which actually describes the directed differentiation of human IPS to the functional ovarian granulosa-like cells via transcription factor overexpression. I mean, this was a really great paper. It also demonstrated the utility of this custom type of ovarian organoid, or ovaroid, as you like to call it, as a co-culture system, right? So let's start there first. Walk us through that paper from the Church Lab and how this is one of the foundations for the work that's going on at Gameto.
Yeah, so this work is started by one of the leading authors is Christian Krem, who is our CSO in the company. And with his colleagues, basically, they were trying to figure out this protocol of how to make this what we call now ovarian support cells, but they are actually very similar to granulosa cells, so we can also call granulosa-like cells. And in the Church Lab, there is a team of people using transcription factors to direct the differentiation, as you mentioned, which is actually a different approach than everything that I have done in my life. So most of my scientific life, I was trying to mimic development. So with the transcription factors, we were basically directly bringing the cells into that new state, right? So they started with a pool of different transcription factors and genes that could be potentially responsible for leading these cells into this granulosa-like cell phenotype. And then they came up with, after the screening, with a group of few transcription factors that could efficiently generate these granulosa-like cells. With these granulosa-like cells, they also showed not just molecular profiles of them, comparing them also with fetal cells, but they also look at the functionality of the cells. Perhaps part of the most interesting of that seminal paper was that they were able to also show that when they put the cells in a culture dish with other cells, they were able to form these organoids that we call also ovaroids that have structures that look like a lot the follicular from in vivo. And that also respond to hormones exactly like or very similarly to what it happens in the body. So that was a very exciting proof of principle that we could use that strategy to make these granulosa-like cells. With that, and then I think now that's when the science at Gameto started and the exciting part. Now, how can we use that to help treating fertility, to help improving maturation of eggs, for example? So as Dina mentioned, in our proof of principle work that was published also in Human Reproduction paper, we use these granulosa-like cells that we generated in the lab, taking iPS cells as initial as a starting material, and then we were able to co-culture the cells with oocytes, immature oocytes that had been retrieved. And we were able to show in this paper that the rates of oocyte maturation were increased compared to the standard IVM, in vitro maturation protocols. And also very interesting from that paper, we were able to also show that after using these eggs for fertilization and embryo formation, the rate of euploid embryo formation was also higher than our controls, and actually very similar, at least with published data from current IVF. So this paper was the idea that proved the idea that we could use the cells with the intent of doing in vitro maturation and leading to high quality embryos through this euploid embryo formation. And then along with that, I think another part of the challenge that we had was we were using a cell line that was a research cell line. Now we also had the challenge of translating that protocol into something that could be brought to clinics. And then we have a paper that it's still under revision, but we have a version on bioarchives in which we do a deeper characterization in the cells, single cell RNA sequencing of multiple batches. And during this process, we were also optimizing our protocol, so substituting reagents by higher quality reagents. We also had the protocol that was based in a substrate of matrigel, which poses a huge risk for translating this to the clinics, not just from being derived from animals, but also from being extract from tumors, and therefore there's a huge lot-to-lot variation. So we had to identify the best substrate to move forward. And that was also very interesting to see that depending on the substrate, we were inducing this differentiation. Despite being a very directed differentiation, very efficient and very straightforward, the state of the cells that we were getting after this process was different depending on the substrate we were having. So the substrates were having an additional effect to the transcription factors into directing. So basically with vitronectin, we were having cells that we were calling early granulosa-like cells. And when we differentiate on top of laminin, the molecular profile of the cells was a lot more mature. And we also showed that after all these changes to use better quality reagents, the reproducibility of the protocol was also improved. So this was something that in the work at Paula Arlotta's lab, it was huge. We were always looking for models that were reproducible and we could improve the protocols to allow for that. And that was something that we also saw. So now we are in a good place with our differentiation protocol, with the research that we translate from George Church Lab to start manufacturing the cells in GMP. And as I said, we are keeping everything to the highest quality that we can, the highest grade, and ready to start translation of this into clinics.
Yeah, I mean, and this is that's a nice synthesis of all the like the nitty gritty stuff that people don't think about that you need to deal with. You need to get all through those hurdles in order to make a product that can be incorporated into human practice. And this is a product. Well, the Fertilo. I don't want to pigeonhole Gameto. I think the scope is much broader in terms of like long-term vision, as Dina you described there a bit. And I'm sure we can come back around and can tell us some more. But in terms of the scope of Fertilo itself, like it's remarkably broad, right? And I want to be very clear here. Like there are a lot of indications where like there's an emergent medical need, like cancer patients. I got to start my chemo, right? I need abbreviated stim. Also, like PCO patients. I think that's a big class, a large class of patients who could benefit from this and really reduce their risk profile. But I would say that a lot of the scope there are women who are undergoing egg freezing to preserve fertility for so-called this isn't my term, so-called social, not medical indications, social egg freezing as it's called. Right. Some people don't like that term. I get it. And because there's like really important needs, right? That's an unmet need as well. Social egg freezing. It's not like, I think I'll freeze my eggs. Like a lot of patients really need to do this. All right. So I want to be clear on that. But I think that a large part of what's driving the growth, what has driven the growth of Gameto, which has been pretty remarkable over the last couple of years, multiple oversubscribed fundraising rounds, which I think reflects like a really high expectations for commercial success on the part of the market and the investors. And as I said, I think a large part of those expectations are driven by that segment of the target population, that's so-called social egg freezing. And while that need is no less real, I want to be clear there again. It's different, as I alluded to earlier, from the kind of cell-based therapy perspective, where we're no longer talking about like a curative therapy, like you need islets because you don't have any islets, right? Islets versus insulin, which is like a therapy that's working. It's worked for decades, maybe 100 years. Anyway, so there's a different kind of rationale there. I don't want to call it a social application of cell-based therapies. And of course, again, I want to kind of underscore that I think it's an inevitable extension of an important extension of stem cell-based therapies. But right now, again, it feels new. So I looked in the committee there. You have this bioethics committee among your leadership, some really high-level bioethicists. Is it the newness like that? Is that why you have these bioethicists so prominently placed? I'll put it to you, Dina. And also, like, extend that, like, our bioethics, this is real, germline and reproduction, and there's a lot tied up in there. Are there other like bioethical questions that come to the fore when you're trying to chart the scientific direction and culture of Gameto? Dina, you first.
Yeah, I mean, you've said a lot in there. There's a lot to unpack. First one, with your social egg freezing, I'd call it more of like a societal impact. As you know, globally, we have a decline in population, and because of all sorts of factors, we're no longer having children when we used to. So, whether you call it social or societal existence, I'd argue the latter, but it's a huge and important impact. And we've seen this is a global phenomenon, and we've seen governments around the world trying to subsidize these treatments. Since 2021, Japan, again, a leader in stem cells and iPSCs, has started to provide subsidies for up to 6 embryo transfers for women to undergo IVF treatments. There's even a pilot in the Kanto Authority, where Tokyo is, to give women financial loans to freeze their eggs, because we know that if you freeze eggs younger, you have a higher success rate later on when you do IVF treatment. So I would say that perhaps, the first part is that societally we're realizing that we need access to assisted reproduction so that people can form families they want to when they want to do so. And there's an also component that of health equity, where right now 1 in 5 heterosexual couples suffer from infertility. Perhaps even more same-sex couples rely on science to build their families, but only about 2% of babies in the US are born with IVF. And IVF is very expensive. There are only a few, still only a few fertility specialized clinic. There are only a few physicians who have finished this reproductive endocrinology fellowship who are allowed to provide this care, way fewer than obstetrics gynecology doctors. So it's really something that is very long, medicalized, and only available for a minority of patients. So the way I think about it, by making it what Fertilo has the potential, and our really mission is to try and use science to ensure that more people are able to use it, because it's important for the society. Whether you call it social, I call it societal. In terms of the bioethics, I'm a physician by background, and we have there a patient. I think it's just doing good research, and that goes to your endpoints questions. From the moment we've started, we were very much building this in the clinic. My co-founder and chairman of Gameto is the founder and chairman of Inception Prelude, one of the largest fertility clinic chains. They are essentially an end customer of something like this, if it is to be successful. And we wanted to ensure that we build this for the end customer as well, because they need to use it. This is not a direct-to-consumer product. This is not a pill. This is something that has to fit in the workflow of the clinic. So we worked with clinics. We worked with patients. So we had studies all across the United States and in Europe and in Latin America where women would donate their oocytes. So that's why we formed from the very beginning, like a bioethics committee as well, because we wanted to have a patient representative to tell us, like, what do you think of this? Right. And we were very fortunate to have work with Leslie Schrock, who is brilliant in kind of bringing popular science, in bringing science to people who may not have, an MD or a PhD, but are going through these treatments, and their opinion matters. So she's the author of 2 books, one on pregnancy, one on fertility. Herself has been, a patient in reproductive care, publicly talked about multiple miscarriages and other issues around like a woman navigating reproductive care. And she has been instrumental in helping us, in addition to some of the other lawyers and professors that we have on it. But the reason why we wanted to have the bioethics committee is to ensure that the patient voice is there from the beginning. That is another thing that is very important and fully engraved in Gameto's mission. And that is that currently, let's say the burden of IVF treatments, even though, sometimes it's a problem of female biology, sometimes a problem of male biology. So today, if let's say it's a male factor infertility issue, the treatment is still heavily on the burden is still heavily on the woman's body. You would probably treat it with ICSI, intracytoplasmic sperm injection, but it is the woman that has to inject at least 2 weeks per cycle, potentially multiple cycles. The median number per baby is about 3. So we felt that, as I guess, like, we have a mix of talent in the company, both women and men working passionately on this topic, that we should provide that patient perspective, and that we should hopefully build treatments that use science, but also empower patients. And that is another burden, that is another mission of Gameto, because you mentioned, there are some people for this medically, there's some people who may need it. And you're right, like PCOS patients, cancer patients, this is where you're talking about either risk of hyperstimulation syndrome or inability to start life-saving chemotherapy while preserving fertility, respectively. But, I'd argue even further to say that even though it's not necessarily a life-threatening and it's not medically critically, why would you not have a better female patient experience for women that are just doing these treatments for other reasons? So that is kind of why we're really working on reducing that stimulation, making that patient journey better. And it goes by working with patients from day one, so that as you're designing this, just like we're working with clinics, we're working with patients, we're working with regulators, because you're navigating quite a lot of different areas. There's a regulatory uncertainty in IVF, there's patient experience, there's how do you get this integrated into these fertility clinics who ultimately need to service. So it's just because of this additional complexity. But, we didn't go into this because it was easy. We went because we thought it would be easier. So that's where we are.
Well, you guys have certainly really made so much progress, and it's really impressive. Not only the as Bruna described, all the technical hurdles that you overcame, but the deep thinking that you've done and the rationale there as you present it is very compelling. So I just want to applaud you on that. Before we move on to, because fertility, that's what you got going on. That's really, you've advanced with all those efforts, pipeline wise, the most advanced, I would say.
Yeah, absolutely. And that's where Bruna, I think, one of the challenge when we talked about the first application, right? So it's easier, it's in a dish, the level of evidence you need to provide is in vitro, it's cheaper to show that it works in humans than showing it if it's an in vivo therapy. But some of the other issues that we've had, as Bruna mentioned, was how do you set up GMP manufacturing? How do you remove batch to batch variability? And our first cell line from Harvard was restricted for research use. And as she mentioned, it had matrigel from a mouse tumor. Who wants anything with their baby about it? So Bruna's team, when Bruna joined the company, completely re-engineered that. And that actually, I think it's an issue that anybody else translating in stem cell biology has to overcome or has had to overcome. So definitely, like, even though it's a different niche, more in vitro, easier, let's say, to achieve those clinical endpoints, there were still some of the challenges of translation as in any other stem cell company.
And there's also, your vision. Sticking with Bruna, maybe you could elaborate on this. But while Fertilo is really advanced clinically, your long-term visions include tox screening, these organoid, ovarioid avatars for disease modeling or tox screening, and specifically something you just had a pretty significant award that you announced that it was one to support research on these cell-based hormone replacement organoids, I guess they will be, for treating menopause. The second platform I describe here, the amino, is really exciting. And as I said, you just announced a substantial award supporting it, without, of course, betraying any trade secrets here. I just want to get an idea of what do you need to do to deploy a cell-based approach for hormone replacement? What does that look like? And is the technological gains you made in the process with Fertilo, are those applying toward that amino effort? Is there a lot of overlap in these projects?
100%. So I think the beauty of our platform is that it really allows us to use for our different programs. So Fertilo was the first one that we moved forward, and we are very excited about the results of Fertilo. But having the opportunity of using that same platform with other goals, as you mentioned, DOVO, which is our organoid drug screening platform, but most importantly, Amino, as you mentioned, which is a more traditional cell therapy based. And I like to think at Amino kind of similar to what is happening with the islets in the diabetes field that we all are very familiar with, multiple clinical trials in there. So there are patients already that undergo and take insulin. And the whole idea behind substituting that by the islets is that the islets can be part of that conversation in the body and substitute the need of the patient to be taking insulin. I started with this example because I think the listeners are probably more familiar with that, but the goal of Amino would be very similar to this. So once the woman goes through insufficiency of their ovaries, let's say during menopause, for example, now we can use the cells that we make on a dish, leveraging the same platform that we developed for Fertilo. Of course, we're going to have different challenges because now we are talking about introducing them into the body of the patient. But the whole idea is that these cells are going to become part of the conversation with the brain to allow that restoration of the hormones that are missing and improve adverse effects that are consequence of this ovarian insufficiency and several others that we know that appear as a consequence of ovarian insufficiency. We have bone decay, we have cardiovascular conditions, and several other conditions that are more and more being associated with menopause, for example, that we hope to, with this new technology, be also reducing the chance of being developed.
Yeah, quickly, I just want to say it's just a representative of the tremendous scope and ambition of Gameto. It's not enough. I mean, I don't want to make it like sound disparaging. I'm impressed. Like you're going after one thing that's like really realizable, broad scope, and then oh yeah, also menopause. We're gonna fix that for you too. So it's takes a lot of gumption, a lot of courage. And to you, Dina, here's a quick little follow up that I love this gem. It reminded me of how very famously the physician scientist Barry Marshall drank a culture of H. pylori bacteria to prove that they caused ulcers. He ended up winning a Nobel Prize for that. In a similarly daring commitment to your science, Tina, you included yourself among an early cohort of women that underwent the Fertilo procedure. What a like unique and amazing position to be in, to be able to contribute to the data set, but also perhaps I would argue maybe a demonstration of leadership, faith in the technology. Tell me a little more. What motivated that? I can't imagine it was just a matter of like increasing the end value, right?
Wow. Well, this is the biggest compliment I've got so far. I would probably recommend compare my participation more so as like in medical school when we learned how to put cannulas. We did it first on each other. And I still remember when my colleague from class put it in my biceps very much up, and I wasn't sure if I'll be able to move my arm for the next 2 weeks. But essentially, that is really the approach. Like every time we would learn a clinical skill, or at least most of the time, excluding some catheterizations, we would kind of try and do it on each other, and that's how we've learned in medicine. And the idea was, so that's kind of where it come from. So as I went through kind of science, I always wanted to see it. Secondly, as a physician, I've seen that, I think in particularly elective procedures that we recommend lightly, and I'm going back to your kind of social element, when it's medically necessary or not. Sometimes I think we're biased on like the life-threatening, and we don't see maybe the morbidity of some of these elective treatments. So I personally actually came up with the concept of Fertilo when I was looking into my own egg freezing. And I thought, wow, this is so hard. And, I don't respond well to hormones. And, I don't think I'll be able to do this with all the jobs that I was working on. I've just moved to the United States back then. And I was like, this can't be, like made for purpose. And I'm sure more women are having these problems, and maybe they would like to freeze their eggs or, undergo IVF treatment, but this is just really hard to do. It's hard on your body, and to fit into your everyday lifestyle. And it's not only just about the weeks of injections after for a few weeks until your ovaries settle, because you're at risk of torsion from enlarged ovaries with multiple follicles. You also have restrictions on some exercise and your travel and other things that are minor, but we're talking again like months of your normal quality of life, right? So that's where I felt that there's a need for improvement. So the reason why I wanted to undergo our trial, I need to, and I think everybody needs to, in order to really excel, you need to believe in what you're doing. So I wanted to see, like, is the Fertilo cycle that we are pitching, that I need to go and tell you, yeah, this is like a better product, better patient experience, is it really? So I wanted to go through it, and I wanted to convince it for myself. And that's why I did it. And I can say that, again, I have an honest perspective right now. Again, it's a medical procedure. I mean, it is not for everyone. And it comes with risks, even the Fertilo shortened cycles. And people need to understand the risks and benefits and only do it if it's the right decision for them. But I can say that it was much easier to integrate into the everyday life. I only, half day off work. And I really believe that it's something that particularly, busy women juggling are more likely to be able to do and repeat, because unfortunately, this is also a lot about a repetitions game. And as we talked about, it's about 3 cycles, but the more cycles you have, the more likely you are to succeed. So by making that cycle shorter and safer and easier to do, you're more likely to eventually end up with a baby. Nobody does this for fun of doing a cycle. People want something at the end. So truly, the reason I wanted to go through it was to understand a patient perspective and convince myself that what we're building is really a better experience and a sufficient benefit and innovation to all the key stakeholders in the system. And that's why I chose to do it. And I still, think that that's very valuable experience that I've had.
Wow. Well, you're all in, right? You're fully and personally invested in the Fertilo technology. And thank you for sharing that story. That's pretty exciting. And, thank you for both of you for sharing the work that you're doing here at Gameto. And before we let you go, as you know, Bruna, as a listener of the show, we'd like to ask our guests about their journey, right? Like how they got to this point, how you guys ended up at the positions that you are with Gameto. So we'll start things off, just because we don't often have biotech founders or industry scientists here on the show. I was hoping if you could tell us about how you got to your current role at Gameta. So Dina, if you want to start things off, maybe you could talk about your role from going from medical training, like you're alluding to ultimately founding a biotech startup in Gameta. And then Bruna, after that, if you could talk about your road as a stem cell biologist with a very productive training in the Arlotta lab, like we alluded to, highlighted by that first author Nature paper, and ultimately to your current role as VP. So perhaps Dina, if you could start things off.
Yeah, well, I was born in Serbia, and my dad was still is a professor of embryology and histology, so very much sciencey family. I was growing up while he was doing research on like chorionic villi of the placenta, so fell in love with science and medicine at the early age. Where I come from, really, biotech industry doesn't exist, so I didn't know, how that you can like go and start this. So I go to medical school. I went to medical school in the UK. Um, at University College London. And in my second year of medical training, I go into this program, which is Founders Forum, the F-Factor competition. It was like F-Factor, but for STEM. And that's where I get exposed to venture capital and company buildings. I've learned the word venture capital at that time. I remember Googling it, like, what is this, venture capital, the company that everybody works for at this event. And that's when I realized that is kind of my passion. I felt that with medicine, I've learned about everything that can possibly go wrong, but I just had only very few things that I could do about them, apart from penicillins, maybe some of these new mAbs, good fluids. I felt sometimes that my hands were tied, and I wanted to kind of go early and prevent disease from happening. And I had all these ideas to solve problems. So that's kind of like in the middle of my training, I realized that biotech is probably where I'd like to go. But maybe even before going to medicine, I really didn't know these options exist. I didn't know that that's a career path you could follow, because where I come from, it doesn't exist really as much. So after that, I did finish medicine. I did like a first year residency, or F1 academic F1 training, as it's called differently in the UK. I got more and more involved in industry careers. I kept my academic research while also working on venture creation, advising venture fund, later on climbing up that ladder, becoming a partner at Salt, working on early stage biotech, as well as kind of late crossover rounds, life sciences content creation. And that's really how I got to co-found Gameto. I also invested in the seed round of Gameto from Salt Fund. I thank my partners at Salt, AJ and Alex. And also I had the great opportunity to work with Martin, who is himself a prolific entrepreneur, 5-time unicorn founder of 7 children, 1 grandkid, who's been a great mentor to me as I've been building the company over the past years. And then after that, obviously, bring incredible people like Christian, Bruna, and other scientists we have in the company. So I'd say kind of going from always academic medicine, always being that person who wants to do research, not necessarily, that much front-facing clinical work, then going and kind of trying to learn on the job basics of finance, corporate, legal, and then going from biotech venture capital into company creation. If I knew always I wanted to go into science and medicine. I didn't really know I'm going to end up here, but I feel very, very happy and very fortunate about it. And every day I wake up and I feel very happy that I'm in the United States of America, where I could live this life. So I try to make the most out of it.
And I'm also not from the US. I guess you probably realize that by now. So I come from Brazil, and where I did most of my training. I always knew I was going to be a scientist. I was in between being an astronaut, paleontologist, and I ended up in biology. And I was actually very lucky. My first year in the Federal University of Rio, I met this new professor who had just came back from his postdoc at the Scripps. And he was really interested in using stem cells to study diversity of the neurons in the brain. And I was just first year as an undergrad in Brazil, thought that was very exciting. And that moment, I'm sure you guys were probably like getting into the field, already in the field as well. But like the cool thing, the challenge was how to make this cell turn into a neuron or into a cardiomyocyte or so on. That was the big question. And that's what I was working with. Because he was one of the pioneers of stem cells in Brazil, I got to leverage a lot. Stevens, I owe a lot to him. So he turned the lab into the National Lab of Stem Cells, and we were training researchers in Brazil, Latin America, along with Professor also Lígia Pereira in São Paulo. And that really empowered me as a stem cell scientist, seeing the field growing. During my PhD, I got a fellowship to spend 1 year abroad. And then I came to the lab of Derek Rossi at Harvard Medical School. That was also the first time that I saw science leaving the lab and going to a company. So for the ones that don't know, Derek has founded multiple companies. And I was like, wow, that's cool. Like, it's in Brazil, I also wasn't exposed to that. So it was interesting, but also driven by all this entrepreneur vibes that Derek had, he ended up phasing out of academia. And that's when I went to the lab of Paula Arlotta. Paula, on the other hand, although she has her entrepreneur side, she's very academic, very fundamental biology. And I had a blast in her lab. But I started thinking back again about the academic career path. So we're very deep into how the brain is made, how conditions associated to the brain are there. So at that moment, I was kind of like bouncing around. That was when COVID hit as well. But at the same time, as someone who has been going to the ISSCR since the early 2000s, I've been seeing more and more stem cells get into clinics and starting to have a real impact. So when in the beginning, there was a lot of hope and we were managing that hope with the society, now I was really seeing that the cells were coming out and doing what we had been promising for several years they were going to do. And I wanted to be part of that. So I wanted to be in one of these companies supporting this development of these cells into bringing changing patients' life. And that's when I saw Gameto, and I was very excited about the science they were doing. Also thought it was very unique. And I decided that I could apply my expertise into women's health. At the same time, I was like personally going through with my partner, thinking about IVF and how to have a baby. And I think everything aligned well. And I decided to make this move to industry.
Wow. Thank you guys, both of you, for sharing those journeys. So unique, but a lot of common elements, and not just, your international origins, but I think a shared ambition toward this next chapter in the translation of stem cell science and the entrepreneurship that is, of course, going to be attached to that. Two unique paths that I think also reflect this unique entry of a major contender into the stem cell based therapeutic marketplace. And we've really enjoyed hearing the science, sure, but more than that, I think what you guys brought is a unique perspective that I think a lot of our listeners are gonna appreciate. We don't get so often on this show, so really appreciate you guys. Really excited to see everything you guys got coming down the pike. I mean, it's really like I said before, your ambition is only matched by your amazing, dedication and technical prowess. Searching for adjectives here, but there's just too many. You guys are great, and it's been a great chat. Thanks again for joining us.
Thank you so much.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast or by email at info@stemcellpodcast.com, where you can give us feedback or suggest guests. Thank you guys so much for listening. Until the next episode, you should tune into some of our previous episodes, which are brilliant. if I do say so myself.